everyone. I'm Allyson Gage from Cohen Veterans Bioscience, and I'm very happy to be here today to share some stories with you. And I'd really like to thank the Precision Evolution Group for allowing this to happen. And I'm sure, as many people have said over the past day and a half, I am really looking forward to doing this again in person, maybe later this year or early next year. So what I want to talk about is how CVB is trying to set the stage for researchers, whether these are researchers in industry, academia, or government, to have validated biomarkers in CNS disorders to move the science forward much more quickly for clinical use in clinical trials and eventually also for diagnostics. And we focus in this area because there are so few other organizations that think about psychiatry and in particular trauma-related brain disorders. So think PTSD, TBI, and suicide. And you can see that when you look at what we call at CVB our brain trauma scorecard. And we look at this every year to see how things have changed to also measure what the impact of our work is potentially doing down the road. And so for PTSD, there are no approved diagnostics, so everything is done based on symptom-based reporting, and it's a very heterogeneous population. There have been 2 approved therapeutics. These were 19 years ago, so nothing in even the remote past. And they are both the same mechanism. They are both SSRIs. And for those familiar with the field, PTSD used to be in the anxiety category in the DSM, and antidepressants were often sort of borrowed over from depression indications and tested in all anxiety indications. And so that's how that happened. There was no purpose, biological-driven strategy for that. In TBI, there are 6 approved diagnostics, but these are aids in diagnosis. So, for example, a blood test that aligns with CT findings, but they still need to rely on symptom-based reporting in order to actually make the diagnosis along with these blood tests. So still quite a ways to go in this area. And so CVB focuses on building platforms to help fast-track precision diagnostics and tailored therapeutics so we can move away from our current symptom-based nosology and move towards more mechanism-based approaches. And if you look at the example symptom list for PTSD on this slide, you can see it's extremely heterogeneous. And if you look at the individual symptoms, you might not be sure if you're looking at PTSD or any one of a number of psychiatric or other CNS disorders. And so CVB has focused the majority of our resources in building platforms to discover, validate, and implement biomarkers into clinical trials to help move the field forward much more quickly than they have been. And there are many categories of biomarkers. We tend to focus on those that could eventually become a diagnostic for the population, an objective diagnostic, or something that actually indicates the course the individual patient might be on, which can help stratify for clinical trials, or something that actually helps predict how somebody would respond to a particular treatment given, whether it's small drugs, biologics, brain stimulation, et cetera. And the goal really is to get to tailored treatments. And so if you start with, on the left, a clinically diagnosed but very, very heterogeneous population like you have in PTSD, what we do is take an approach of applying deep phenotyping across multiple modalities of biomarker assessments. And by using computational methods, whether you're using supervised learning or unsupervised learning, eventually getting to different subtypes of these patients. So taking that one clinically diagnosed population and doing clustering methods to get to different subtypes. And the biomarkers from that multimodal approach that get us there would then be potentially diagnostic biomarkers for these individual clusters. And then taking it a step further, still using our computational modeling to then identify the differences in the underlying biological mechanisms between these clusters, which allows you to then find solutions that target those specific biological mechanisms. So you can now diagnose somebody as a subtype and then pair them with a treatment that is tailored to that biological mechanism. And so we focus on a few different biomarker categories. We look at the omics category, which is mostly measures of specific biological mechanisms, so proteomics, metabolomics, transcriptomics, et cetera. Also, neurophysiological markers. And one of those, EEG, is one of the case studies that I'll present in a moment, but we also look at things like startle reflex, MRI, and then a large category of wearable and home sensor technologies, which I think is really an exciting area that has a lot of promise for psychiatry to discover and validate biomarkers going forward. So, as I said, I'm going to present two different use cases that CVB is involved in moving these biomarkers forward. The first came out of a study that CVB funded, and Stanford was one of the sites, and a professor there, Dr. Amit Etkin, discovered on MRI a connectivity signal that differentiated the PTSD population that was being studied into two different subtypes, and we call them subtype 1 and subtype 2. What was interesting is these two different subtypes, these two different signals seen on MRI, could not be discovered by looking at the symptom reports. So in essence, these people look the same based on their symptom reports, but MRI put them into two different groups. And what these groups linked to functionally were that one of them responded to psychotherapy and the other subtype did not. So that's an exciting finding to move the field forward. But again, this is MRI, so it's not very scalable. Even if you're thinking clinical trials, it's very difficult to implement in multi-site clinical trials for standardization reasons and data harmonization. And then if you want to move it into the clinic, that's not really feasible. And so what CVB did was try to support this to move forward to something more scalable. And we worked with Dr. Etkin to move it to EEG to attempt to find the same signal, even though we don't know if the signal in each of them means the exact same thing, but in essence to use EEG to find different subtypes. And what Dr. Etkin did, along with others, was to find this signal in EEG and test it in different psychiatric populations of PTSD and major depressive disorder. And I'll just quickly show you the results from that. So if you focus on the upper right panel here, this is looking at the PTSD studies that were done. And this upper right one are the healthy controls that were enrolled in the study. And you see in the frontal lobe, in the upper left corner of this box, that there was high activity there. And then when you look at the PTSD population, which is represented on the bottom, there was a separation, again, into two different subtypes. One that looked very much like the healthy controls, So that's subtype 1. You see, again, the higher activity in the frontal lobe. And then subtype 2, which had this dysregulated functional connectivity. And again, in EEG, just like in MRI, these two different subtypes could not be separated based on their symptom reports. They found the same thing in in two different major depressive populations. So this time in the figures, we're looking at the map of the difference between subtype 1 and subtype 2. And so you can see both here. In the first study, again, you saw the higher activity in subtype 1 in the frontal lobes and this dysregulated functional connectivity in subtype 2. And that was seen also in the second major depressive population. So for all four of these studies, what did that mean functionally? Did you see the same thing that you saw in MRI? So we'll take it one population at a time. This is looking at the PTSD studies. And if you look at this first graph, what you're seeing, the blue line is subtype 1. This is the one that looks like the healthy control on the EEG signal. And then subtype 2 of the PTSD population is the one that has that dysregulated functional connectivity. And what you see is a nice separation measuring based on the CAP score after therapy. You see that subtype 1 responds to psychotherapy and subtype 2 does not. The study was not powered to look at the two different types of psychotherapy that were used, but they did do post hoc analyses and saw that there was a trend towards separation, showing that this biomarker could be generalized across, different types of psychotherapy. In the first MDD cohort, instead of looking at psychotherapy, they were looking at this EEG signal to be able to predict those who would respond to drug treatment, in this case, sertraline, a drug approved for major depressive disorder. And they found that subtype 1 responded to psychotherapy more than those that did not receive sertraline, but the subtype 2 group did not. So this signal seems to be generalizable across more than just psychotherapy as a predictor of response. The second depression study was done in one looking for this EEG signal to be a predictor of response to repeated transcranial magnetic stimulation. So a neuromodulation solution instead of drug therapy. In this instance, the EEG signal did not predict response to treatment. And so it's interesting, we might need to look for different types of signals for perhaps different categories of treatment, but it also tells us a little bit about the underlying biology. So it still moves the field forward in understanding the disorders. And it just may be that maybe RTMS actually changes the EEG signal. And so CVB, while EEG is much more scalable, still clinics do not all have EEG technology. And in terms of moving it forward as a diagnostic, we would want to have something even more scalable than that. And so in our device lab, we're beginning to look at the various consumer devices and see whether the signal is seen on these sort of more simple versions of EEG so that it is also more easily incorporated into clinical trials and decentralized clinical trials that someone can use at home. So in the second use case for wearable and home sensor technology, we took a different approach. Instead of finding, discovering, and reproducing the biomarker and then thinking about how to bring it into clinical trials later, here we're starting at the other end. We are trying to determine what's feasible to put into decentralized clinical trials and then begin to discover the biomarkers that come out of the data sets from those trials. And as I'm sure you're all aware, doing these decentralized trials really allows you to get much more data on these individuals. So for us, deep phenotyping in a passive way with these devices and getting much more consistent and frequent data in a home environment, so something that's more natural to the person than, for example, when you're thinking about sleep, somebody sleeping in a sleep lab. There are differences in the sleep patterns of somebody sleeping at home than somebody sleeping in a sleep lab with all of the multiple things attached to them. And so in the next few slides, I'll just show you how we think about the type of data that comes from these devices and whether they can be used in this fashion. And we look at bed sensors, wearables, the wrist and ring devices, and then the headbands for mobile polysomnography. So in the wrist and ring category, the raw signal that comes in for almost all of the devices is accelerometry and PPG, gyroscopes, you get temperature, and then through data processing and pipelines, you then get the biometric signal, which, for example, is heart rate, there are raw movement, skin temperature. You get some context in terms of environmental light. And then those, through your data analysis pipelines, can become the clinical outcomes, your derived outcomes for sleep. So think about sleep stages, the time spent in bed, the time awake after they fall asleep, and their movement, which in PTSD could be very relevant towards thinking about nightmares. When you look at bed sensors, or which I have a picture here on the right of one of the maps that we use, you're now complementing this information that you get from the wearables, and not just having a different look, but also potentially adding that data together to get a better signal. And so with these devices, you get respiration rate, you get interbeat intervals and associated low and high frequency metrics. And this really gives you, when you think about the ultimate outcome, better sleep times, because you get, for example, an idea of when somebody is actually in the bed and out of the bed, which you might not know if you're wearing a wrist wearable or the ring. And then on the headband side, you're getting the EEG metrics, you're getting an idea of their position in bed. And here is really better metrics of respiration rate and interbeat intervals. And now you can really get not just your epics telling you whether they are asleep or they are awake, but actually looking at the different stages of sleeping. So getting much closer to the gold standard of polysomnography. And then you can't forget that while you're busy testing and validating these devices, there are always new devices coming out. And they either have new assessments that they're able to do, or they've improved on the measures. And so you have to constantly assess these and think about what is your ultimate goal and which of these suits your purposes. And in the end, while your best assessment may come from combining all of these different devices, you cannot do that in a home setting. It's hard enough to do it in a sleep lab. We just finished working on a technical validation performance study where we actually had people wearing 6 different devices. And that was complicated enough, but that's somebody who's coming in for 2 nights of sleep, and they can tolerate that. And it was really just to measure the data. It wasn't to really understand the change in their sleep based on some kind of treatment. So CVB had to decide what is our criteria for the different devices, and anyone who takes these on will need to determine their own criteria. For us, it was understanding what is the most reliable data, and that means understanding, can we get the raw data and can we analyze it ourselves so we know we're comparing apples to apples? And so we looked as to whether the data are kept with the manufacturer, and we just get back some analyzed outcome without understanding how that was done. Or from the resource side, sometimes you can get the raw data, but you have to create your own API in order to bring it in. So we went through this in our device lab and our analytics team to understand what are the best options for us. After doing that, we then built essentially our platform for decentralized trials, which we call Trial in a Box. And this essentially is a series of proof of concept, decentralized clinical trials where we are working directly with the participants who have consented and passed screening, and we are just mailing them everything they need for the trial. So they get the devices, they get instructions, and the return information to send the devices back to us. And what we're focusing on in these proof of concepts are two areas that are important strategically to CVB, which are sleep, because it's an important component of both TBI and PTSD, but also across brain disorders, as well as movement. And a part of our criteria is to be able to collect at least 2 weeks of data, and then a certain amount of data per day. And that depends on which indication we're actually looking at, how much daily data we need. And then we also want to compare the objective data we're getting from the devices with subjective outcomes. In sleep, if you're familiar with the field, there's often a difference between polysomnography data and what's self-reported. So our objective is to eventually replace subjective outcomes with the derived outcomes that come from the devices, but also to understand the relationship between other self-reported outcomes and the objective data. And then most importantly, or at least importantly, to be able to fully automate this process and be able to get within 48 hours of the data being generated, a dashboard of clinically relevant outcomes back to the person that's evaluating the data. So eventually back to the patient, if that's the use case, or back to a physician, if that's the use case. We have built what we call the study manager in order to do this full automation. And so we have an automated ingestion of all of these data types, automated standardization of the data and processing, and then finally automated data analytics so that we can get this dashboard back to someone within 48 hours. Now, this can depend on the participant in the study. So, for example, for some of the wearables, the person actually has to click a button to say that they're uploading the data to the cloud. For other devices, it's automatically uploaded, and that isn't a factor. And so our first trial in a box is a sleep proof of concept, and it is fully decentralized. And our main goal is to understand the feasibility. So just because we understand our instructions of how to set up the devices doesn't mean the individual we're going to mail it to will understand that. And that's really what this is. Can they get this box? Can they use the devices properly? Will they charge them as they're supposed to? Will they fill out the information, the questionnaires that pop up on the app? So in this box, in this study, they get one wrist device, one sleep mat, they get a phone with a study app, and all of the instructions we think they need to move along. And here we're collecting 4 weeks of data. We don't need that much data, but we'd also like to understand from this study when is there a fall off in the person's compliance with using the devices or answering the questionnaires that are also included. And so, as I mentioned, we are comparing the derived sleep outcomes from the devices with the subjective report. So in this case, it's a sleep diary, asking them, how long did it take you to fall asleep? How much time did you sleep, et cetera. We're also collecting other ECOA related to sleep and ecological momentary assessments and seeing how that aligns with the objective data. And again, doing this, processing this data through our pipelines within 48 hours. And so our goal is for this platform to enable us to look at the data and discover biomarkers for sleep and eventually validate them, but also to have this platform for others to use in their clinical trials going forward. Hopefully, the next time we talk, I'll be able to explain how feasible this is in a decentralized environment, clinical trial environment, and I look forward to any questions that you might have.